Hey everyone, Professor Davis here, and we are going to be diving into the cellular basis of inheritance. And this particular chapter, we're going to really kind of look at meiosis and that process of inheritance of chromosomes to the next generation. And so meiosis is part of that idea of sexual reproduction. So meiosis is a type of cell division, and it reduces the number of chromosomes by half. This is why, again, it's represented by N instead of 2N. And the purpose of meiosis is to form structures that we call gametes, or cells we call gametes. This process of creating the male gamete, which is sperm, is called spermatogenesis. Remember, genesis means the beginning or the start of sperm in this particular case, and then oogenesis, which is the production of eggs. So you have spermatogenesis and oogenesis. Now, gametes are the reproductive cells that are haploid in their number, and these are known as sperm and ova. Another word for ova is egg. These are made through the process of meiosis. Now, it's not just good enough to make egg and sperm. In order to make a new individual through sexual reproduction, those gametes have to restore the chromosome number back to 2N, and this process is known as fertilization. So the process that restores the number of chromosomes to the diploid number, the 2N, when fusion of egg and sperm nuclei happen, they produce a cell called a zygote, which is now again a 2N cell, where half the chromosomes came from the sperm and half the chromosomes came from the egg. Now, this process of meiosis and fertilization is known as sexual reproduction. So sexual reproduction has to involve the two processes of meiosis and fertilization. So meiosis, guys, the purpose again is to produce gametes, sex cells. And so we start first with a germ cell. These are located in the ovaries in females and the testes in males. This is that 2N germ cell is going to then divide. It divides and then divides again. And this is what's key. This is what makes it different than actual mitosis. It's going to divide twice. And because it divides twice, ultimately it can have 4 cells that come out instead of 2. And each of those cells has half the chromosomes. So we say that it has N chromosomes. Okay, so it has N. These are going to be the gametes, whether they're egg or sperm. Remember, egg is formed through the process of oogenesis, whereas sperm is created through spermatogenesis. So guys, in males, this is spermatogenesis. This is the formation of sperm. This does take place in the testes, and it does create those haploid sperm cells. So if you look here in this particular diagram, it shows you that the cells are going to divide. They divide through meiosis I, meiosis II, and at the end of meiosis II, they have half the chromosomes. Okay, so each of these sperm in this particular case, the cell started with 4 at the top. They have half the chromosomes, which means each sperm cell has 2 chromosomes. Okay, now in humans, they would start with 46, and each sperm would then have 23. All right, and you'll also notice that it creates 4 sperm by the end. Okay, this is as known as spermatogenesis. Now in females, the process looks a little different. This is oogenesis. Oh, and with spermatogenesis, guys, this starts occurring at puberty, and it continues pretty much the rest of the life of the male. Males can end up producing billions of sperm on a regular basis. All right, and so they are going to create tons of sperm cells, and they're going to see a just slight decline on it as they age. In females, however, the process is a little bit different. This is known as oogenesis, the formation of the egg or ovum. It's in the ovaries. However, women are born with the amount of eggs they're going to have. Okay, so all the eggs they have are already going to be in part of this process of meiosis. They're actually suspended in meiosis, getting ready to divide. And if that egg is chosen that month, it's going to finish its maturing and become the actual egg that gets released during that month. Women release, when they start puberty, they release 1 to 2 eggs every month. That is then going to give them a chance during that cycle to potentially conceive and become pregnant. If not, that egg is then shed and released, and the process starts over again. This continues, however, in females until menopause happens, where then the process stops. This is what makes it different from males between males and females. Another thing here too is, if you'll notice, the division doesn't create 4 eggs at the end. Remember I told you back in the last chapter that that process of dividing the cytoplasm is not always equal? And we see that sometimes a bigger cell may form than the other. Well, this is true here in oogenesis. In each of the divisions that occurs, one cell is going to be larger, and that one continues the process of creating that ovum. And only one egg comes out successful. The other ones are known as polar bodies, and they just get pushed off to the side and recycled. The reason we need that one egg to be larger is that all the chromosomes are present, in that sense of the 23 that come from mom, but so are all the organelles. So every organelle that you have in your body cells came from your mom. So the mitochondria, the Golgi, the rough ER, the smooth ER, all those came from your mom because they were in the egg, whereas the sperm only delivered the chromosomes. Okay, so all those organelles came from the egg. That's why they are larger than the sperm cells. It's also why only one is released at that particular time. Okay, and so these are a little bit of the differences between the processes of oogenesis and spermatogenesis in males and females. All right, so this right here kind of maps out what fertilization looks like. So we saw with meiosis and how it divided and it created these in cells. So we have an egg cell here, and then we have also a sperm cell. These two are then going to fuse together for fertilization, and when N plus N comes together, we get the 2N. This is the first cell after fertilization, and this is the new organism. This is the new individual because they now have restored the 46 chromosomes when we talk about this with humans. And this is also what makes this zygote in this individual very unique, is because it's a unique combination of 23 chromosomes from mom and 23 chromosomes from dad. Now, if meiosis does not occur prior to fertilization, the zygotes would contain 2 times the number of chromosomes than the parent. And so if you started out with 46 chromosomes, if we didn't do meiosis before fertilization happened, the new organism would have 92. And then if those 2 organisms then didn't do meiosis first and you had 92 each, then obviously when you would add those together, you'd have 184 and so on. The chromosome number would get out of control. And so because of that, we need to make sure meiosis happens before, halving the number of chromosomes before fertilization can happen. All right, so let's look at the sexual life cycle when we look at animals. This is a diploid life cycle. Now, life cycles vary depending on which organisms we're looking at, whether they're animals, plants, fungus, that sort of thing. So if we're looking here in the human body, though, or even the animals, when we talk about any kind of diploid life cycle for animals, we have a male and female. Each of them are 2N. However, in the testes and the ovaries, these processes of spermatogenesis and oogenesis do occur, creating sperm and egg. When the sperm and egg come together and fuse through fertilization, this creates a new organism with the zygote. Fusion of egg and sperm create that nuclei that restores the 2N number, and the zygote is formed. This zygote then undergoes mitosis for growth, where we go from 1 cell to 2, 2 to 4, and so on, until the complete new, in this case, human being has developed, whether male or female, and then the process then can continue. And if you'll notice that the only part that actually is a living organism here, when we talk about the idea of a new human, a new bear, a new cat, a new dog, is when fertilization happened and a new zygote was formed. Okay, and so when you look here, that creates that diploid. And so in order for an animal to survive, it has to have that diploid number. It has to have the 23 chromosomes from mom and the 23 chromosomes from dad. Now, in fungi, this is different because fungi live what we call a haploid life cycle, meaning that the fungus that you actually see, like the mushroom or whatever that is going to be the actual haploid number, which means they have half the chromosomes. Okay, they only have half of them. And so then the spores that you see do have new variation that comes together. They do have a small part of their life cycle that's diploid, but most of the time the fungi lives in a haploid life cycle. So it's kind of like the opposite of what we would see for humans or animals. Plants, however, do what we call an alternation of generations. They go back and forth between the two, haploid and diploid. Okay, and depending on which type of plant it is, depends on how long they spend in each of these. But there is still a back and forth that happens between meiosis and fertilization. It just means that parts of the plant are going to be in a different part of that life cycle at a given time. Okay, so this is showing you a fern, and if we were to look at what we would call the Archegonium, which is part in the Antheridium, which are parts of the fern, those organisms or those parts of the organism are haploid. However, the fern that you see actually growing with the leaves and the fiddleheads and all that kind of stuff is diploid. But a part of the plant is still haploid, going through that process of creating spores and then also creating then those gametes. Okay, so they alternate. And you'll learn more about this if you take biology too. All right, so let's go back here and look at chromosomes for a little bit. When we talked about chromosomes, remember that chromosomes, when they get ready to divide, they are going to then be replicated. However, you need to have 2 chromosomes of number 1, you need 2 chromosomes of number 2, 2 chromosomes of number 3, right? If you're a diploid organism, this means when you have 2 of them, they are called homologs. Okay, when we look here, homo means same, and so these mean that these chromosomes look alike. They're going to have the same length, the same centromere position, they're going to have the same genes that are present. And so in this particular case, they picked out a homolog, and they're showing you that on this particular one, eye color is found here. So you have the blue allele that came from your mom, the female, and the brown allele that came from the male, but they're located in the same area, and they both are going to code for eye color. Now, they code the same gene, but if you'll notice, they code for a different color. That is different. Okay, that part doesn't matter as much as long as they both are coding for eye color. That's the part that makes them the same. That's the part that makes them what we call homologs. Now, chromosomes can be separated by size. With pairs being matched, just like we saw with those homologous chromosomes, homologs. And this is known when we look in humans. This is what creates that 23 pairs. Okay, because pair means you have 2. So you have 2 chromosomes number 1 over here, and you have 2 chromosome number 2 over here, 2 of 3, 2 of 4, and so on. And this is what, again, creates those 23 pairs, which equals your 46. Now, the first 22 pairs are what we call autosomes. They're the ones that all humans have in common. We all have the same 22 pairs of autosomes. The 23rd pair is what's different. The 23rd pair is what we call the sex chromosomes. And these are only found in animals, and they are going to be what determines the gender. Okay, so XX is the female, XY is male. These are not considered homologs when you are male because X and Y are not the same. Okay, and so because of this, and this is found again only in animals, we do see that XX is female, XY is the male. Now, autosomes must have 2 per set, but sex chromosomes, you have to have at least 1 X. If at least 1 X is not present, that particular individual is not viable. It will not grow and develop properly. And so you have to have at least 1 X. Okay, now if you have 2, that's XX, that's female. If you have 1 and there's a Y, then that's male. And this is how they look. We do see that we can pair up these chromosomes from large to small. They get paired together based on their homologs. And this process of going through the idea of mapping all of these chromosomes, seeing them out and laid out where every gene is located, is called the Human Genome Project. This started back in 1999, and it took about 11 years for them to map out the human genome. Okay, so it took a very long time to map them out, but now they know that on chromosome number 2 in this particular area is a certain gene. They were able to map those out. Now, this mapping out or a picture of the chromosomes is known as a karyotype. This is showing you the complete set of chromosomes, and they can be arranged in homologs like you see here. So you have the 2 chromosome 1s together, 2, 3, 4, 5, all the way down to 22. Okay, so they have all their homologs when we look at the chromosomes through 22, which are the autosomes. But down there at the bottom, you're going to notice that you have the X's and then you have the Y's. This is the sex chromosomes. So this first one, the first one is a normal human male because it has an X and it has a Y. Now, if you'll notice, this is why they're not homologs, because the X chromosome is a lot larger than the Y chromosome. And there's a number of reasons for this. The X chromosome codes for a lot of other things besides sex, whereas the Y chromosome just pretty much codes for what we call maleness. Okay, and so there is a big difference there. On the other hand, if you look in the second picture, you'll see that there are two X's. If there's two X's, then this is a normal human female. Now, guys, I say normal here because they all have two chromosomes. Okay, there are some abnormalities that can happen where you have more than two chromosomes, or you could have where you don't have the two, and that means that something is going on. And we'll talk a little bit more about those later. And so this is known as a karyotype. All right, so this brings us to the actual process of meiosis. Meiosis, remember, has two divisions instead of one. So we call these Meiosis I and Meiosis II. Those are what the two divisions are. Now, during Meiosis I, homologs are going to separate. So those chromosomes that are similar, like chromosomes 1, will line up together and they're going to separate, that sort of thing. And during Meiosis II, the chromatids are actually going to separate. This causes them to become what we call daughter chromosomes. All right, and so there is a little bit of difference between Meiosis I and Meiosis II, which we're going to take a look at. All right, so this is a little bigger picture here. When we look at the idea of meiosis, we see that the first cell is either going to go from this area or this area. And you can see that there is a either or random arrangement that occurs because this is a random assortment of chromosomes. As they split, okay, so it's an either or, they're not bolting, but as they split, they're going to continue to go through the process of dividing until at the end you have 4 different cells, and they have different sets of chromosomes that are present. Okay, and you can see them in this picture here. They're showing you an either or there. So let's look at interphase first. Now, interphase is going to be very similar to the interphase that we saw in mitosis. There is a G1, there is an S, and there's a G2. And this interphase occurs prior to the cell dividing its first time in meiosis I. Okay, so interphase is very similar, okay? But we do see that there is going to be some differences as we actually get into the divisions that happen in meiosis I and meiosis II. So this diagram just gives you a little closer look where you can see at the top you have prophase I all the way through telophase I. And then you have prophase and you have all the way through telophase in mitosis. And so this just shows you that meiosis I and mitosis are going to see that there are differences that occur. And you can mostly see it based on, especially in metaphase, how the chromosomes line up at first. Okay, so you can see chromosomes are lining up in pairs up there in meiosis, but they're lining up in straight lines over in mitosis. So let's look at why. So in meiosis I, they are going to go through prophase, metaphase, anaphase, and telophase. But we're going to put a 1 with it so that you know that it's happening during meiosis I. So in prophase I, the chromatin do condense into chromosomes. This is exactly the same as what we saw in mitosis. The nucleolus also disappears. The nuclear envelope fragments. And in animal cells, the centrioles migrate to opposite poles. And spindle fiber formation begins. These are all exactly the same of what happens in just normal prophase. Here's the difference. Okay, so there's a little star next to it on purpose there. There is something here that happens. This is called crossing over. Crossing over between what we call homologs, those homologous chromosomes, is going to occur. Okay, so what does this mean? Crossing over is when homologs undergo what we call synapsis. They come together and they actually exchange, share, switch genetic information. And this only occurs during prophase of meiosis I. That's it. It's the only time this happens. So here's my 2 chromosomes. Okay, I had gotten one from mom and one from dad previously, so I marked them with 2 separate colors here. Remember that during interphase, they replicate. See how they replicated? All right, and they create this kind of like X-like structure. In this idea of crossing over, these homologs, these 2 chromosomes that have the same information on them, just I got some from mom and some from dad, they're going to come together here. And this is what creates the synapsis. Do you see how the X's like have pieces that are crossing, like the blue is crossing with the kind of pinkish color. There's crossing that's taking place. In those areas where they cross, they're going to actually exchange the genetic information. So if we were to pull these apart, this is now what they would look like. So some of the chromosomes are a mix of mom and a mix of dad. Guys, this is what allows us to have so much genetic variation in the human population. This is what allows us to have so many differences in our genome, which is actually a good thing. Okay, is this idea of crossing over, exchanging genetic information. Now, when the 4 chromosomes come together like this, in that sense of the 2 X's coming together, these are known as tetrads, because tetra means 4. These tetrads are going to be a term used during prophase and metaphase of Meiosis I only. You only see these structures in Meiosis I, prophase I, and metaphase I. Okay, so here's the homologs, and they come together like this to create the tetrads. Okay, so when they're like this is when they're known as tetrads. All right, so now you can see the tetrads on here, and they were present in the prophase picture, but now we're in metaphase. In metaphase, the homologs or tetrads randomly align at the metaphyseal plate, the equator. And they undergo this through what we call independent assortment. They're not going to care where all the moms are lining up or the dads are lining up. They're all just lining up in the middle. Spindle fiber formation then is going to be complete. They've connected into these kinetochores here. Okay, so metaphase 1. In anaphase 1, these tetrads, these homologs, are going to separate. Do you see how we are separating those homologs? These replicated chromosomes migrate to opposite poles, and cytokinesis is going to begin. So guys, it's very similar to what we saw in mitosis, but those tetrads make it a little different. Now, chromosomes here are going to then unwind into chromatin. In telophase 1, the nucleolus reappears, the nuclear envelope also rebuilds, and cytokinesis is almost complete until it is. Now I already have 2 cells, okay? But guys, if you'll notice here, I still have 4 chromosomes that are in there in this particular case. Started with 4, I still have 4, okay, when we look at this. The thing is I don't have 4 all in a line like we saw there. I've got 2 over here and 2 over here. Okay. So I've got these chromosomes that have been present. All right, guys. So if you'll notice here, the chromosome number's already been halved. I started out with 4 chromosomes earlier, but now I'm down to just 2. The thing is though these 2 are already still replicated. And so because of this, we're not going to enter into a normal interphase again. The stage that happens between telophase 1 and prophase 2 is known as what we call interkinesis. So by the end of Meiosis I, the number of chromosomes have already been reduced by half. And this is where you count those centromeres, the center parts. Chromosomes, though, are still going to be in their replicated state because another cell division is going to occur. And this is what we call interkinesis. We are not going to replicate DNA in this stage. So there's going to be a G1 and a G2, but no S, because S is the synthesis of DNA. We're not doing that. So we have a G1 and a G2, but there's no S. So we call this interkinesis. This then allows us to go into Meiosis II. Okay, so no S. We already have enough DNA, so different from interphase. So in Meiosis II, prophase 2, the chromatin condenses. The nucleolus disappears. The nuclear envelope fragments. The centrioles migrate to opposite poles. And spindle fiber formation begins. Because if you'll notice, this isn't one cell doing this, there's now 2. Okay, so now there's 2 cells going through this process. In metaphase, the replicated chromosomes or the sister chromatids align at the metaphase plate or the equator. And spindle fiber formation is complete. So again, this looks very similar to mitosis. The issue though is I have 2 cells going through this, and you'll notice that there's mixtures of chromosomes here. In anaphase 2, the chromosomes are going to come apart because the centromere splits. Single chromosomes migrate to opposite poles and cytokinesis begins. Again, though, 2 cells going through this, Anaphase II. Telophase II, chromosomes unwind. They unwind into what we call chromatin. The nucleolus reappears, the nuclear envelope rebuilds, and cytokinesis is almost complete. And now I have, instead of 2 cells that were identical to the parent, I have 4 unique cells that have half the chromosomes from the parent. As if the parent started with 4, there's now only 2 in each of these cells. So we went from 4 to 2. In humans, it would have gone from 46 to 23. All right, so this is showing you meiosis II. 2 different cells are going through this, prophase 1, prophase 2, metaphase 2, anaphase 2, and telophase 2. Now, guys, this is what leads us to what we call genetic variation. Sexual reproduction ensures that there's genetic recombination and variability in the offspring. Okay, this is really important, okay, in this process. So this idea is known as the Red Queen hypothesis, and it has to do with the fact that it's not just the organism, it's also the stresses on the organism based on their environment, their diet, all these different things that help them decide how to reproduce, when to reproduce. And the whole idea too is this recombination and variability that happens allows for the species to be greater at surviving when outside forces from the environment come in. Okay, so this again is why it's really important that genetic recombination does occur when we're talking about this idea of new pathogens that potentially come in, new things that are environmental that we want to help protect our species against. And what helps in this whole process is that we do have crossing over that takes place. This allows for different combinations of genes on those chromosomes. And remember, this takes place during prophase 1. There's also something called independent assortment, and this is where the tetrads randomly align during their metaphase, allowing again for different combinations where you might have different combinations of mom and dad's chromosomes going into different egg and sperm, which makes each of them very, very much unique. And then, of course, fertilization is also random in the sense of what sperm actually fertilizes what egg. And so because of these 3 things, crossing over, independent assortment, and fertilization, this allows for so many combinations of our genetics. So here's a little note to kind of put it into perspective in a number. This allows, when we talk about those 3 things, this allows for a possibility for a possible 4.95 times 10 to the 27 genetically different combinations of genes / chromosomes for each zygote of a single couple. So 2 people who come together and they decide that they're going to have children. There is this many different combinations that could occur based on their genetics. And guys, this is the number with all the zeros. The 4.95 times 10 to the 27. It is a huge number. So this doesn't mean, like, it doesn't matter how many kids that these 2 people have together, they are never going to be the same. Kid 1 versus kid 30 are going to be totally different unless they're identical twins. That's different. If they're identical, this means they came from the same egg and the same sperm, so their genetics are the same. But we're just talking about general siblings. So you may have certain characteristics that are similar to your siblings, but you are still going to be completely unique from any of your other siblings. Okay, because even if you came from the same parents, because there's so many different combinations that can occur. This is also why you may look like one parent more than the other. Or at certain parts of your life, you may resemble more than the other. Okay, I have a brother that I look very much alike. We have a lot of similar characteristics because we got a lot of them from our dad's side. We have another brother who got more of my mom's family side of the genetics, and so he looks a little different than us. He has more blonde hair, whereas Ryan and I have brown hair, dark brown hair, for that matter. Okay, but when we're together, you can start to see that there are certain similarities that are similar. I also have, my daughter looks a lot more like my brother that doesn't really look like me. She ended up getting some more of those combinations from my mom's side of the family. And so this is where you can end up seeing it. This is also why certain genetics, which we're going to talk about in a little bit when we get to genetics, where some stuff can stay hidden, where all of a sudden a redhead comes out of the mix and you're like, where did that come from? But if you go back to through your lines, somebody in the family had red hair and now it's reemerging based on the combination that occurred. And so these are those advantages. So advantages of sexual reproduction is it provides a lot of genetic variation. This does help increase the chance of survival of some offspring, not all. Okay, but it does increase the chance for some, and that's the whole point when we talk about success in nature. Survival of the fittest is not only to survive yourself, but to pass your genes on to the next generation and they survive, and so on. It creates that idea of a legacy. That's what makes something successful in nature. All right, so let's do a quick comparison between mitosis and meiosis. So mitosis, remember, is associated with asexual reproduction. So these produce daughter cells that are genetically identical, okay? We see that in meiosis, it's associated with sexual reproduction, and this produces haploid daughter cells that have N chromosomes, and it allows for variability. So mitosis leads to stability, meiosis leads to variability, okay? So there's a difference in their purpose. Mitosis occurs in your stem cells, also known as your body cells or somatic cells. Meiosis occurs in your germ cells, which are the sex organs, testes, and ovaries. Mitosis has one nuclear division, whereas meiosis has two. And mitosis produces only two daughter cells, where meiosis produces four. The exception there, though, is oogenesis in animals, egg formation. So the exception there is oogenesis, or egg formation. All right, so guys, again, here is showing you meiosis, showing you Meiosis I and Meiosis II. Also, it gives you a picture of mitosis. Mitosis is more similar to Meiosis II, so that's why they moved it over. And again, in words, it shows you some of the differences, the process in meiosis versus mitosis. So this could be a good reference slide for you if you want to kind of stop and pause the video so that you can really kind of compare and contrast mitosis and meiosis. All right, so this brings us to chromosomal abnormalities. How do alleles interact? Why is this a big issue? Different alleles arise through mutation. This is actually rare. A lot of them are more stable, and they are inherited changes in the genetic material. Most mutations are actually beneficial. However, the ones we normally hear about are the ones that are detrimental, the ones that cause problems. Now, these mutations are not the ones that cause us to be like X-Men and things like that, which would be kind of cool if we had those kind of powers, but we don't. But they do allow us to have a lot of different variety. So guys, they call the wild type, when we're looking at the allele that's present in most of the populations. The other allele a lot of times is known as the mutant allele, the one that's different. Okay, so the wild type is the allele present in most of the population, whereas the other allele is the mutant allele. A genetic locus is what we call polymorphic if the wild type allele is present less than 99% of the time. The occurrence of more than one form or morph of the phenotype in a population. And this is what we can see in a lot of things when we talk about the human population, is that we have a lot of mixtures of forms, where you're going to end up seeing lots of different eye colors, lots of different skin colors, lots of different colored hair. That's natural. Okay. When we look at this, and it gives lots of different forms to it. So guys, a mutation is a permanent change in a gene that's passed on to an offspring. Now, we're going to talk about this briefly here, but it's going to come back when we get a little deeper into genetics. And so we do see that there could be a change in actual chromosome number. The changes in the DNA number may be genetically engineered, like we see a lot of times with plants, but they also can occur accidentally during meiosis. So in plants, guys, we learned about haploid and diploid, but in some plants we call them polyploid because they have more than 2 copies of chromosomes. If they have more than 2 chromosome sets for each set, they could have 3. They're called triploid if they have 3 of each set, of each group. If they're going to have 4 in each of their sets, they're called tetraploid. These are seen only in plants. We do not see this in animals, or especially in humans, but we do see it in plants. And this is actually how a lot of the genetically modified plants are. When you think of strawberries at the grocery store, this is what we're talking about. The strawberries you find in nature are very small compared to the strawberries you find at the store. They're very large. The ones that you get at the store have been genetically engineered, and they are normally going to be tetraploid, meaning they have 4 sets of chromosomes instead of the 2 that the normal strawberry had. It didn't mean that we gave it like elephant DNA or anything like that. It just means it has more of its own strawberry DNA, which makes them larger. Okay, so this is what we see in plants. In animals, however, we say that they have an abnormal or uneven chromosome number. This is called an aneuploid. This is where they have one extra chromosome or one missing chromosome. It's not an actual whole set. It's just they have one missing or one extra. And so monosomy is shown here. Do you see how you have two for each, but in one of them they're missing one? That's called a monosomy. Mono means one. Trisomy, on the other hand, is when you have two for all of them, but one of them has three. All right, and that's where the tri comes from for the three. So guys, when we look at a polyploid, when we're talking about with plants, these are due to genetic manipulation. Offspring may be sterile because of this genetic manipulation. But the purpose in plants and stuff that we see with this is that they are to create seedless plants. Since they're sterile, they don't actually create seeds. So when we talk about seedless watermelon, that's what we're talking about here. We've actually created this in order to make it where it doesn't have the seeds. Also, when you do polyploid plants, it makes them hardier plants. They tend to be able to withstand changes in the environment a little bit better. Also, certain ones have more attractive flowers. And so that can be something that's beneficial. And a lot of them are known as super plants because they grow larger than normal, like I was talking about with that strawberry. On the aneuploid side, when we talk about for animals, this is due to non-disjunction. So we talked about the chromosome. When it looks like this, it's supposed to pull apart, right, where one goes one way and one goes the other. In non-disjunction, it means when the cells are dividing, it goes one direction. So this cell over here gets 2 copies. This cell got 0 copies. Okay, and so there's a mismatch that's going to occur. Most monosomies are lethal, especially the autosomal. If they only have one chromosome 1 or only one chromosome 15 or only one chromosome 22, those are lethal. They are not going to survive that. Trisomies are usually detrimental, meaning there's going to be some health issues, but they're more likely to survive with that. Survivors usually suffer from distinctive sets of physical and mental abnormalities, and we call them syndromes a lot of times. You've probably heard of like Down syndrome. It's a trisomy where you have 3 copies. Okay, and that's 3 copies of chromosome 21. And so this is why it gets that term as a syndrome. Not all Down syndrome people have all the same kind of physical and mental abnormalities, but they do contain certain ones. So when you have a change in the chromosome, so these are all examples when the chromosome number is messed up. Let's talk about if the chromosome itself is kind of damaged in some way. A change in chromosome structure could be due to things like radiation, UV light, chemicals, viruses, and the chromosomes can break and fragment, and they usually repair properly, but sometimes they don't. And if they do not rejoin properly, a mutation in that chromosome might occur. And so one example of this is inversion. This is where a piece of the chromosome rejoins, but it rejoins in the reversed or upside down order or direction, which I'll show you a picture of in just a minute. We also see that translocation can happen where a piece of a chromosome rejoins the wrong chromosome. It's an unrelated non-homologous chromosome, so it joins somewhere else that it shouldn't have, and that's going to mess up both chromosomes when it does that. You could have a complete loss of the piece of chromosome. That's called deletion. We've lost it. We don't even know where it went. And if a segment in the chromosome is repeated, we call that a duplication. Now, if this occurs, guys, in the germ cells or during gamete formation, like the formation of egg and sperm, the mutation is passed down to the offspring. Okay, if this happens as you're an adult, just like in your skin cells, it's going to cause potentially like cancer, but it's not going to be passed on to your children. The mutation has to be present in your egg or sperm in order for it to be passed on to your children. And guys, Down syndrome is mostly a trisomy, which we just talked about, but in 5% of the cases, it's actually due to a translocation that has occurred. All right, it's not in all cases, but in 5%, it's due to translocation. All right, so guys, this is what those mutations look like. A deletion, you'll see that A was just deleted. This makes the chromosome shorter, which means you're missing some of the genes. Duplication, you can see that D and E were duplicated. There's an extra part. It makes the chromosome longer, but it also messes up the code. We also see an inversion where C and D broke off, but then they reattached upside down. And then you have translocation is showing you that Q and R ended up breaking off, and so did G and H, but they switched places when they rejoined. They rejoined to the wrong chromosome. So this shows you some examples. Now guys, I'm going to come back and talk to you more about mutations a little later in a different video, but I wanted you to see that in this idea of meiosis, as these chromosomes are being pulled apart and stuff like that, and that formation of egg and sperm, mistakes can happen. All right, and if these mistakes do happen, if they occur, these mutations do happen, then the effects do affect the new offspring, the new individual. Okay, so this just is that kind of intro into genetics through the process of meiosis. All right, if you have any questions or concerns, please let me know.